Bismillah Rahman Rahim. We start another paper 4, which is the latest exam, November 2024. It's the question paper 4-2. As you all know, this paper is for 2 hours, but I mean, it's not enough time for it. So you must, number one, read the whole question. Number two, you must do the questions that you know very well. If you don't know any question not very well, just leave it and come back to it. I would like you to do a paper worth 80 marks and get 60 out of 100. If you do 80 marks and leave out 20 marks, I'm okay with it. But as long as you're getting 60 out of 100, it's an A grade. So the grade boundaries are anything between 60 and 65 is an A grade. Starting with question number 1, figure 1.1 is a diagram of part of a Bowman's capsule and a glomerular capillary. Identify structures A and B. This is a podocyte, and the B is the capillary endothelial cell. Capillary endothelial cell. So the capillary has endothelium. That's the only place where we call it endothelium. In the blood vessels, the endothelium is present in all the arteries, veins, and capillary. In the capillary, it's only the endothelium. So capillary was, of course, in brackets, but endothelial cell was the answer for it. Then it says the glomerular filtrate is produced in the Bowman's capsule by the process of ultrafiltration. State the conditions required for ultrafiltration. Look at the marks, 2 marks. So be careful in writing it. What is the main 2 points? There has to be a higher hydrostatic pressure or higher hydrostatic blood pressure. And has to be a basement membrane which acts as a filter, so only small molecules can pass through the basement membrane. So state the conditions required for ultrafiltration. So basically there has to be the only question when you answer this is to think of it, what would go wrong? Why would ultrafiltration not occur? Well, it would not occur when there is not high hydrostatic pressure, and that happens when a patient comes in the emergency with a lot of bleeding and the volume of blood has decreased. That reduces the pressure of the blood, so kidneys fail. So the person had an accident and is losing blood, but his kidneys will go on to shut down. And that is the first thing we do in the emergency is that we set up a drip so that we can restore the volume of blood. As the newly formed glomerular filtrate passes along the proximal convoluted tubule, selective reabsorption takes place. The fluid remaining in the proximal convoluted tubule eventually forms urine. Table 1.1 lists 3 of the components of blood plasma that enter the glomerulus in a healthy person. Complete table 1.1 by writing increased if the component is present and is higher in concentration than in the blood plasma. Decreased if the component is present and lower in concentration than the blood plasma. Same if the component is present is of the same concentration and not present. So you have to either say increase, decrease, same, or not present. You may use each response once, more than once, or not at all. So glucose, the component in the newly formed glomerular filtrate, is the same. Component in the urine is not present. All the glucose that is filtered is going to be reabsorbed. And in diabetics, why there's glucose in the urine is because there's too much glucose. The insulin is not being produced. When the too much of the glucose is filtered, well, there's a V max in which maximum can be absorbed. And that is it can't take in the extra glucose. That glucose is not reabsorbed and that is passed out in the urine, but that only happens in diabetics. Large plasma proteins component not present, not present. Plasma proteins are too large to cross the basement membrane. So if they are not filtered out, they cannot be present in the urine. And if, God forbid, they are present in a person, we find out it's because the basement membrane is damaged. And that is a disease which is called glomerulonephritis. Now, urea in the filtrate is the same. And it will be increased. Why will it be increased? Because this urea in the blood is in 5 liters of blood. But the volume of urine is maybe only 200 milliliters or 300 milliliters. So the same urea now in a smaller volume of water it will be more concentrated. The reason being the urea same. It's just like there's 10 teaspoons of sugar in a big jug of water, and now the same 10 teaspoons of sugar in half a glass of water. So it will be increased. Part C says sweating during exercise can lead to a response in the posterior pituitary gland and in the kidney. State the response to sweating that occurs in the posterior pituitary gland and the response that occurs in the kidney. The posterior pituitary gland will release more ADH. You see, the posterior pituitary gland will release more ADH because now the blood is too concentrated. The person has lost a lot of water in the sweat. And the kidneys will reabsorb more water. Or you can say urine become more concentrated, or you can even say lower volume of urine. Or you can give an explanation as the more aquaporins added to the membrane in the collecting ducts. So the collecting ducts becomes more permeable to water. So you have to give me the response in the posterior pituitary gland and the response in the kidney. So better if you give these sort of headings like this. Posterior pituitary gland does what and kidney does what. Yeah, even if you don't give the heading, but you think like that, because you have to get your 2 marks. Remember, you have to get your 2 marks. Now, coming to question number 2, this is a very direct question, and either you know it and you should give me all the points in a point form. Please don't write long paragraphs in which doesn't make any sense. Outline the processes that may, it says several different processes can affect allele frequency in populations. Outline the processes that may affect allele frequency in wildlife populations. So the first thing is mutation, then genetic drift, then founder effect, then bottleneck effect, then natural selection, or you can even say directional or disruptive selection, then migration, or interbreeding between populations, then genetic recombinations, sometimes linkages, groups are broken and crossing over. So all these points, as long as you have given them to me in uh, in a proper way, I mean, I'm not saying just write it in points. Don't write it in paragraphs. Just give me the points. Why would allele frequencies change? Why would suddenly sickle cell anemia be more common in a certain country? Why? Because everybody there is the ones who are homozygous are disadvantaged because of malaria. So you can give me all those points, and you need to be just, if you don't know this question, then just skip it and go on to the next question, which you know very well. Now, as you can see, this is for 6 marks. And then some of the points which you can write in any of these questions in which they talk about how allele frequency is affected. Like mutation is one you can write everywhere. Then natural and directional selection you can write everywhere. So all these points are something which are very common to all these questions. And if these ever come in your exams, these are the points you can always give. Then coming to part B of the same question, the process of selective breeding changes allele frequency in a population. Plant breeders use selective breeding to improve crop plants such as maize. Explain how selective breeding can be used to obtain a variety of maize where the plants are vigorous and of uniform height. Now, when this is inbreeding, results in a lot of homozygous plants and there's increase in homozygosity. So what you do is you do outbreeding. That means you produce a hybrid. So you cross a weak plant which is producing less crop yield with one which is high yielding, and then you get these hybrids and the offsprings are heterozygous, so those are going to be increasing the crop yield. So inbreeding produces results in a lot of homozygous plants, and this of course reduces the crop yield. And so if you do outbreeding, outbreeding means you cross a weak a crop, a plant which has been homozygous for, which has been inbreeding for many years. That's called inbreeding depression, with the one which is not been not has been bred, not has been self-pollinated, thus has more higher crop yield. So you cross them, this results in a hybrid. These have increased heterozygosity, so this is going to increase the crop yield. Now, coming to question number 3, the treatment of many diseases has been improved by the use of genetic technology in medicine. Explain what is meant by genetic engineering. 3 marks. What is genetic engineering? The manipulation of genetic material. You take the gene or the allele and transfer it into another organism. And then you ensure that the expression of the gene takes place. Like, for instance, you take the insulin gene, then you take out the plasmid from a bacteria, and then you cut the plasmid, and then you add the gene for insulin, and then you join the plasmid and you get recombinant DNA, and then you put this plasmid into another bacteria, and then you culture the bacteria, and then you ensure that the insulin gene is switched on, so insulin will be produced. So basically, you're talking of gene editing or producing genetically modified organisms, which are called GMOs. So a phenotype will be modified or a named protein will be produced, like insulin will be produced. You can give me the example of it also, or you can write it in general terms as well. So as long as you get your 3 out of 3 in this question. So manipulation of genetic material is genetic engineering or genetic technology, and gene of interest transferred into an organism, then you ensure that the expression of the gene takes place and a named protein is produced, and this results in the production of genetically modified organisms. Part B1, Leber congenital amaurosis LCA is an inherited eye disease. In LCA, the photoreceptor cells in the retina die at an early age. This causes impaired vision, reduced eyesight in children, which can progress to blindness. Mutations in different genes cause different forms of LCA. One form of this disease, LCA2, is caused by mutation in the RPE65 gene. Gene therapy has been used to treat LCA2. Outline how an inherited eye disease such as LCA2 is treated with gene therapy. So you add the healthy gene or the allele to the virus, and then you inject the virus into the eye. The gene integrated into eye cell DNA, and then this gene is expressed to produce the functioning protein, and we use an adeno-associated virus AAV, which of course is given in your book, so you could talk about that as well, but you would get your 3 marks by saying that you add the healthy gene, you inject the virus into the eye, then the gene is integrated into the eye DNA, the cells of the eye, the cells of the DNA of the eye cells, and then the gene is expressed to produce the functioning protein. So you add the healthy gene to a virus, then you inject the virus into the eye, then the gene is integrated into the eye cell genome, and the gene is expressed to produce the functioning protein. Part 3 suggests why the eye is a suitable organ for gene therapy. It's easily accessible, easy to inject, low risk of an immune response, and eyes small in size, so small amount of gene therapy treatment needed. So easy to inject, low risk of immune response, and small amount of gene therapy needed. Part C, LCA10 is a different form of LCA caused by a recessive mutation in the CEP290 gene. This gene codes for the protein CEP290, which is involved in the correct functioning of photoreceptor cells in the retina. The mutation of CEP290 causes an error to be made when the primary transcript is spliced to form messenger RNA. The abnormal mRNA that is formed has an extra sequence of RNA nucleotides known as exon X between exon 26 and exon 27. Exon X contains a stop codon. Figure 3.1 compares the effect of mutation in CEP290 with the normal gene expression. So the normal is this exon, and there's intron, and there's 27. And in section of in the LCA10, the exon 26, and then there is a mutation here. In the normal 26 and 27, there's an intron. In the 26 and 27, there's a mutation here in the primary transcript. Now, when the mRNA is made in the normal thing, the 26 and the 27 combine. Here we have a problem. The 26, the X, and the 27. So shortened SCP because it becomes a stop codon. So shortened CEP290 polypeptide, and this is the normal CEP290 polypeptide. Now here it's a shortened polypeptide. In 2022, research was carried out into possible treatment of LCA10 using genetic technology. A human clinical trial investigated the treatment of LCA10 using a short RNA nucleotide sequence known as cepofarsen. Cepofarsen binds to the section of the primary transcript containing the CEP290 mutation, so that normal splicing occurs and functional CEP290 protein is synthesized. Not this one, but the normal one is synthesized. People in the clinical trials received regular treatment with ciprofloxacin to the eye with the greatest loss of vision, treated eye for a period of 12 months. Changes in the light perception, visual acuity of both eyes are measured using a vision chart. A negative change in the visual acuity score shows an improvement in visual activity. So a negative change in the visual activity score shows an improvement in the visual activity. Figure 3.2 shows the results of the clinical trial over 12 months. Whenever you have to read such a lot of information, please read it twice and then look at the graph and then sort of explain it to yourself. So the key was eye not treated and the eye treated. So this one, the eye was not treated. This one is the one it was not treated, and this is the one which was treated. So one with the excess. So impaired activity, we had change in visual activity, improved acuity. Impaired acuity is 0.1, 0.1.2, and then improved is 0.7, zero point 0.8, and all that. So you look at this very carefully. You look at the time in months, and it's done for 12 months. Then we come to the question, it says describe. Describe, please remember, is no biology. Describe the results of the clinical trial data shown in figure 3.2. Let's look at the data now. Now, when you're looking at the data, please remember there are two or three things that you need to remember. Number one, do you see a pattern? Do you see in the pattern there is something abnormal? Then you write about that. Now, generally you look at both the graphs. Now, in both the graphs, what do you see? Improvement in acuity of both eyes. Or okay, you don't say that. You say overall visual acuity of both eyes becomes more negative. You see, from 0, this went to -1, and this from 0 went to minus 0.5. So overall acuity of both eyes becomes more negative. So that was an improvement, because it said the more negative it is, the more it is improvement. Now look at the difference. Treated eye, this was the treated eye. Which one was the treated eye? This one was the treated eye. You can see it here, the eye treated. So the treated eye has more improved acuity, or you can say has more negative visual acuity. It went from 0 to point somewhere here, some figure here, point between 0.5 and 0.6. And then there was an anomalous reading at 9 months. It does not fit the pattern. You see, this is generally increasing. You see, it's increasing, it's increasing, it's increasing. It's increasing, the negativity is increasing. I mean, the graph is like this. And then there is this at 9 months. There is an anomalous result at 9 months. Does not fit the pattern. And then you could give me a data code. Data code means value of one eye at 2 months or values of both eye at 1 month. So you could have given me value here, the value here of the eye not treated, and the value here of the eye at 5 months, or anything. I'm not saying definitely you have to say this. The mark scheme is very categoric. It said values of one eye at 2 months, or you say values of both eyes at 1 month. So you could have given me the values at 5, you could have given me the value at 6 or 7 or whatever you wanted to give. That is called a data code. So you give me figures from the graph. And that would have got you your 3 out of 3 of the described. So both eyes become more negative, treated eye more negative acuity than the treated eye has more improved or has more negative acuity than the other one. Then there's a anomalous result in 9. And out of all these data codes, you could have only given me one. I'm not saying you have to give me all 3 of them. You have to give me any one of them. Then coming to the last part of this question, another method of being investigated pre-LCA10 is to use gene editing tool known as CRISPR-Cas9 system. The CRISPR-Cas9 system uses a short length of RNA called guide RNA. Guide RNA is complementary to the target DNA, is a link to a nuclease enzyme called Cas9. Cas9 breaks phosphodiester bonds in DNA. The cell repair mechanism repairs the cut in DNA after the modification has taken place. A vector delivers Cas9 and two specific guide RNAs to the photoreceptor cells. They act on the section of DNA which contains the mutation. Exon X is no longer added to the mRNA. Explain how this method used to treat LCA10 is an example of gene editing. You see, the mutated DNA is deleted The mutated DNA is deleted or removed, or the DNA is replaced with normal DNA. And it happens at specific locations between exon 26 and exon 27 in the genome. So the functional protein CAP290 is now made, and it acts on the person's own DNA. So this is an example of gene editing in which we edit the person's own genome. So this will get you your 3 marks. So mutated DNA is deleted between exon 26 and exon 27. Functional protein is made, acts on person's own DNA. And the stop codon is no longer present. So stop codon no longer present. Question number 4. Genetic crosses can be used to investigate patterns of inheritance. A mutation in a gene involved in fruit color in tomato plants. Solanum lycopersicum results in the production of yellow fruits instead of red fruits. A genetic cross goes between a pure breeding plant with a red fruit and a pure breeding plant with a yellow fruit to produce the F1 generation. All F-spring plants produce red fruits. The F1 plants were then crossed with each other and the seeds produced were planted to obtain the F2 generation. Construct a genetic diagram to show the cross of the F1 generation that produced the F2 generation using the symbols R and R. So this would be very simple, RR, because the F1 generation is always heterozygous. And then you show the gametes, you circle them, you get a mark for that. And then you get RR, R small r, R small r, and R small r. So these would all be red, and this would be yellow. So there's 3 marks for that. It's 1 mark for this, 1 mark for this, and 1 mark for this line. Please remember, genetic experiments are marked per line, so you have to get the entire line correct to be able to get that mark for this. So just writing it properly. So the gametes, so this would be parental genotype. This will be gametes, this will be offspring genotype, this will be offspring phenotype. Part B, a theoretical dihybrid cross involves 2 genes located on different autosomes. Each gene has 2 alleles, one dominant and one recessive. Parent homozygous dominant for both genes is crossed with a parent that is homozygous recessive for both genes. So A, D, D, this will be homozygous dominant for both genes is crossed with small a, small a, small d, small d. So crossed with a parent that is homozygous recessive for both genes, this produces F1 individuals that are then crossed to produce the F2 generation. So the F1 generation is all going to be this. So these produce that are then crossed to produce the F2. So this into this. So heterozygous for both is crossed with a heterozygous for both. Now you know the ratio for that is 9:3:3:1 ratio. I've always taught you this and I've always explained this to you again and again in the class. Explanation. What will be the explanation be? Independent assortment of homologous chromosomes in metaphase 1 of meiosis. So you see, you have to give me the ratio that got you 1 mark and then any two from the explanations. If you said independent assortment of homologous chromosomes got you the 2 marks. If you give me further detail of it that this takes place in metaphase 1 of meiosis, well, you got a mark for that. You could also have said metaphase 2 because it happens both in metaphase 1 and metaphase 2. So it could both be right.